These are the lecture notes for Chapter 16, How Populations Evolve. There are 3 sections. Section 16.1 is on genes and populations and how they are, how genetics is related to evolution. Section 2 is on natural selection, and then Section 3 is on how we maintain diversity of living things. So, in Section 1, this is a review from Chapter 1, actually, when we learned about the levels of organization. A population is a group of organisms of the same species that live in a particular area. So if we're talking about members of a population, we have to be referring to all of the members of the same species. We can't say the population of birds. We have to be correct, we have to say the population of the population of I'm sorry, I lost my train of thought. But a specific species of birds, so the population of barn owls, or the population of robins, or the population of cardinals. You know, you have to be very specific on the species. And then when we learn about population genetics, this is the study of the diversity in terms of allele differences. And remember, alleles are versions of a trait. So the differences in alleles among a population. It evaluates the diversity of a population by studying the genotype and phenotype frequencies over time. Now, there's microevolution and macroevolution. Macroevolution leads to the formation of new species. But microevolution pertains to evolutionary changes within populations that just maintain the diversity within that population. Because the more genetic diversity, the more likely that if there is a disease or some kind of natural disaster that wipes out some members, that all of the members of the population will not be destroyed. The more diversity there is, the greater the chance that some of the members of that population would survive. Okay, so below that it says various alleles at all the gene loci in all individuals make up the gene pool of the population. So the gene pool is really all the alleles or all the versions of a trait that there are in that population. So we can describe the gene pool in terms of genotype frequencies and allele frequencies. Okay, here's some examples. You may have, if you've learned biology in the past, if you've taken any kind of biology class and talked about natural selection, more than likely you've heard the story, which is a real, actual story, of what happened with the peppered moths in England during the Industrial Revolution. This is the color of the majority of the peppered moths before the Industrial Revolution. They had this kind of peppery color. They were light colored with some flecks of black. And then you had some mutated moths that were dark colored. Completely dark colored all over. So these were the 2 phenotype versions, was light and dark. Okay, the genotypes would be, since dark happens to be dominant, the genotypes would be either either homozygous for 2 genes for the dark version, the dark pigmented moths, or heterozygous, a big D and a little d. And then the genotype for the light-colored moth is 2 little d's, homozygous recessive. So genotypes can be described as homozygous dominant, heterozygous, or homozygous recessive. Okay? The alleles are 2 big D's, a big D and a little d, or two little d's. And that would be the alleles. As far as when we talk about phenotypes, genotypes, and alleles, that's what we mean. And in this particular case, the natural selection, since there were more light-colored moths, but there were a few dark-colored moths, when the Industrial Revolution happened, and all of the trees that the light-colored moths were living in or on, the bark of those trees that used to be a kind of a shade similar to the light-colored moths turned dark because of all the soot and all the pollution in the air. Then the light-colored moths stood out, they got eaten, and more dark-colored moths began appearing in the next few years. So that was an example of microevolution within the peppered moth population. So allele frequencies. When we talk about allele frequencies, it's the proportion of each allele within a population's gene pool. So the frequency of the big D and the frequency of the little d, for example. Frequencies of the dominant and recessive allele always add up to 1, just like, percents always add up to 100%. So we describe this by an algebraic expression, but it's really easy, so don't get worried. P plus Q equals 1. P is the frequency of the dominant allele, and Q is the frequency of the recessive allele. So we could say, for example, that P and Q always have to be a decimal. So let's say the frequency of the big D allele is 0.3, then the frequency of the little d allele is going to be 0.7. Okay, so microevolution involves a change in P and Q over time. If the gene frequencies don't change over time, then there hasn't been any microevolution. So what we do is we look at P and Q in 1 year, and then we look at the next year at the frequencies of P and Q, and if there's a change, microevolution has occurred. And the greater the change, the greater the extent of the microevolution. This is a part of Hardy-Weinberg equilibrium. So what we're looking at here is where the frequency of big D, or P, equals 0.2, and the frequency of little d, or Q, equals 0.8, then 0.2 and 0.8 is 1. Then we can also use another equation to describe the genotype frequencies, and this would be P squared, which is the frequency of homozygous dominant, plus 2PQ, which is the frequency of heterozygous, plus Q squared, which is the frequency of the homozygous recessives. That also equals 1. So even though this looks like a really complicated algebraic expression, just remember these like P squared just represents the number of homozygous dominant. 2PQ is the number of heterozygous. Q squared is the number of homozygous recessive. So if we know that P is 0.2, then we know P squared is 0.2 times 0.2, which is 0.04. So that means the frequency of the homozygous dominant individuals, that means 2 big Ds, is going to be 0.04. And then we can take 2 times 0.2 times 0.8 to give you 0.32 is the frequency of the heterozygous big D, little d individuals. And then since we know Q is 0.8, we can say 8 times 0.8. This is a mistake. It should be 0.8 times 0.8. That says 0.08, but it should be 0.8 times 0.8 gives you 0.64. And that's the frequency of the heterozygous individuals. So at this point in time, when this information was collected, there were more, not heterozygous, but homozygous recessive. But at this point in time, there were more homozygous recessive, light-colored moths in the population because the Industrial Revolution had not taken them out yet. Okay, for Hardy-Weinberg equilibrium to be true, that means that the frequencies will remain constant, and you should understand that this hardly ever happens. because the environment is constantly changing. There are constantly mutations. I mean, it hardly ever happens. But if a population has reached Hardy-Weinberg equilibrium, then these 5 things must be true. There must be no mutation, no migration, a large gene pool, random mating. In other words, individuals are just mating with each other randomly. They're not choosing mates. And no natural selection is occurring. If those things all happen, then the population frequencies, the allele frequencies won't change. And this is just showing in a Punnett square how to calculate the frequencies of big D, big D, big D, little d, and little d, little d. It's just a different way to represent what we just went over. Now, the required conditions to meet Hardy-Weinberg equilibrium are rarely met. So anytime there is a change or a deviation in the P's and the Q's, that indicates some evolution has taken place. The greater the change, the greater the extent of the evolution. So this shows you that after the natural selection of the dark colored moths, we end up with, you see how the light colored little d moths were 0.64. After the Industrial Revolution, they were 0.04. And there were much higher frequencies of the homozygous dominant and heterozygous individuals. So it's just showing you that microevolution occurred due to the Industrial Revolution. So this is a table that helps you determine if evolution has occurred. First of all, the Hardy-Weinberg condition is random mating. Well, non-random mating would be deviation from the condition. And did evolution occur? No, not according to that, because the moths do mate randomly. And no selection, no selection is the Hardy-Weinberg condition. The deviation is selection. There was definitely natural selection. Birds chose to eat the, this is a terrible picture. This tree should be dark so that the dark moth blends in. So picture that in your mind. But birds were choosing to eat the light-colored moths because they could see them better against the dark trees that were stained from the pollution from the factories. So they were definitely choosing the light-colored moths. They were choosing a particular phenotype. So that is evolution occurred. That says evolution occurred. And you can go through this chart, but all the other conditions were met. All the allele frequencies changed. There was definitely mutation because the dark moths are a mutation to the light-colored moths. Mutation is just a change. And there was definitely a migration in and out of the population. So those different conditions were met. Now, mutation just means a change in the DNA code. So genetic mutations are the raw material for evolutionary change. You can't have evolution without genetic mutations, because how can nature choose the characteristics that are more suited if there aren't different characteristics to choose from? So mutations provide new alleles. Some mutations might be more adaptive than others. In other words, better for survival than others. The mutation of the dark-colored moths was better for survival at that particular time because it made them blend in with the dark trees. Now, gene flow, another word for that is gene migration. That is the movement of alleles between populations, and that can occur in plants if pollen or seeds are carried to another population. And it can occur in animals when individuals migrate into and out of the population. It mentions here the concern of using genetically modified plants, because if you have a plant that's been modified to be resistant to a herbicide, and somehow that gene makes it to into a weed, the herbicide is not going to be able to affect that particular weed anymore. continual gene flow reduces genetic divergence between